The topic of today's discussion is Introduction to a Scientific Inquiry, using coffee beans as an example. Learning outcomes. Formulate specific scientific questions based on research. Design and conduct experiments effectively with accurate data collection. Analyze and interpret data to draw valid conclusions. Communicate findings clearly to various audiences. Cultivate a mindset of curiosity to promote continuous scientific inquiry. Contents. Identify a topic of interest. Conduct preliminary research. Define a specific question. Develop a hypothesis. Choose variables. Design the experiment. Gather materials. Conduct the experiment. Record data. Analyze data. Draw conclusions. Consider implications. Identify limitations. Suggest further research. Communicate results. Seek peer review. Revise the experiment. Reflect on the process. Document everything. Stay curious. Conclusion. Identify a topic of interest. Start by selecting a general area that interests you. This could come from personal experiences, observations, or topics you read about. Consider aspects that intrigue you or where you see potential for exploration. For instance, you might be curious about how different methods affect the flavors of coffee. This initial stage helps set the foundation for your scientific inquiry. A clear area of interest can motivate you to delve deeper into research and experimentation. The key is to focus on something that captures your curiosity and invites further investigation, like the different extraction processes of coffee beans. Conduct preliminary research. Before formulating a specific question, gather background information on your chosen topic. This research helps you understand existing knowledge and identify gaps that your inquiry might fill. You can read scientific articles, books, or credible websites to familiarize yourself with various extraction methods and their effects on flavor. This step is crucial for building a solid foundation and may lead you to refine your question. For example, you might discover different brewing techniques, such as espresso or French press, and their impact on flavor extraction. The goal is to collect relevant information that can guide your next steps. Define a specific question. After researching, narrow your focus to formulate a specific, testable scientific question. A good question should be clear, focused, and researchable within your resources and time constraints. It's essential to identify the exact aspect of your topic you want to explore. For example, instead of asking, "How do you make coffee?" a more specific question could be, "How does the brewing temperature affect the flavor extraction of coffee beans?" This targeted approach allows for a more manageable and meaningful investigation, guiding your research and experimental design. Develop a hypothesis. A hypothesis is a testable prediction based on your research and specific question. It should propose a potential outcome or relationship between variables. A well-formed hypothesis can guide your experimental design and help you focus your inquiry. For instance, you might hypothesize that brewing coffee at higher temperatures will result in a more robust flavor extraction compared to brewing at lower temperatures. Your hypothesis serves as a starting point for your investigation, providing a clear direction for your experiment and a framework for analyzing the results. Choose variables. Identify the variables involved in your experiment. Variables are elements that can change or be controlled during the investigation. Clearly define the independent variable, what you will change, dependent variable, what you will measure, and controlled variables, factors which are kept constant. For example, in your coffee extraction experiment, the independent variable could be the brewing temperature, for example, 85 degrees Celsius, 90 degrees Celsius, 95 degrees Celsius, while the dependent variable would be the flavor intensity assessed through taste tests. Controlled variables might include the type of coffee beans used, the grind size, and the amount of water. Understanding your variables is crucial for a well-structured experiment. Design the experiment. Create a detailed plan outlining how you will conduct the experiment. This design should specify the materials needed, the steps to be followed, and the methods of data collection. A well-structured experiment allows for consistent results and helps avoid confusion. For example, you might outline a procedure for brewing coffee at different temperatures, specifying how long each batch should steep and how you will measure flavor intensity. Having a clear plan ensures that the experiment can be replicated and that the results are valid. Consider potential challenges and how you might address them in your design. Gather materials. Collect all the materials and equipment needed to conduct your experiment. Having everything ready before starting will help the experiment run smoothly and efficiently. List all required items, such as coffee beans, water, a kettle with temperature control, a French press, taste testing cups, and any measuring tools. Ensure that your equipment is in good working condition and suitable for the tasks at hand. Organizing your materials beforehand can save time and minimize errors during the experiment. Being well prepared helps facilitate a more effective and enjoyable research experience. Conduct the experiment. Follow your experimental design to carry out the investigation. Ensure you adhere to the established procedures to maintain consistency and reliability in your results. Carefully brew coffee at the different temperatures you've chosen, keeping all other variables constant. It's crucial to stay organized and methodical during this phase. Document your observations and any changes made during the process. Conducting the experiment according to your plan allows for valid comparisons and insights. If unexpected issues arise, note them for future reference. This step is essential for gathering meaningful data to answer your research question. Record data. Accurately document all observations, measurements, and outcomes during the experiment. This data is vital for analyzing results and drawing conclusions. Create a clear system for recording data, such as tables or charts, to ensure that all information is organized and easy to interpret. For your coffee extraction experiment, note the flavor intensity ratings from taste testers for each brewing temperature. Detailed records help you track your findings and provide a basis for further analysis. Good data collection practices are essential for maintaining the integrity of your research and ensuring replicability. Analyze data. Examine the data collected during your experiment to identify patterns, trends, or correlations. This analysis can involve statistical methods or simple comparisons, depending on the complexity of your data. For instance, compare the flavor intensity scores across different brewing temperatures to see if higher temperatures consistently yield stronger flavors. Look for significant differences or relationships that can help answer your research question. Analyzing your data allows you to draw meaningful conclusions and evaluate whether your hypothesis was supported or refuted. Clear visualization, such as graphs or charts, can enhance understanding and communication of your findings. Communicate results. Effectively share your findings with others through various formats, such as presentations, reports, or publications. Clear communication is crucial for disseminating your research and contributing to the scientific community. Tailor your communication style to your audience, whether it's fellow researchers, coffee enthusiasts, or the general public. For example, present your results at a local coffee science seminar or submit a paper to a relevant journal. Using visuals such as graphs and charts can enhance understanding. Engaging communication helps ensure that your research reaches its intended audience and fosters discussions. Seek peer review. Engage with peers or mentors to review your findings and methodology. Peer feedback can provide valuable insights, helping you identify strengths and weaknesses in your research. Share your results with fellow coffee enthusiasts, scientists, or professors who can offer constructive criticism. This process not only improves the quality and credibility of your work, but also promotes collaboration within the scientific community. Be open to suggestions and willing to make revisions based on feedback. Peer review is an essential step in the scientific process, fostering transparency and enhancing the validity of your research. Draw conclusions. Based on your data analysis, interpret the results to determine if your hypothesis is supported or refuted. Conclusions should reflect the evidence collected and provide insights into your research question. For example, if your analysis shows that higher brewing temperatures result in significantly stronger flavor extractions, you can conclude that temperature has a substantial impact on coffee flavor. Summarize your findings clearly and concisely, highlighting key points. This step is essential for understanding the implications of your research and for communicating your results effectively to others. Consider implications. Reflect on the broader implications of your findings. Think about how your research contributes to existing knowledge in the field and its potential applications. For instance, your results on coffee extraction methods can inform brewing practices for coffee enthusiasts and industry professionals. Discuss how understanding temperature's effect on flavor extraction can enhance coffee quality and consumer experience. This consideration not only adds depth to your research, but also connects it to real-world contexts, demonstrating its relevance. Highlighting implications can lead to further discussions and explorations within the scientific community. Identify limitations. Acknowledge any limitations in your experiment that may have affected the results. Understanding limitations helps clarify the context of your findings and guides future research. For example, if taste testers have varying preferences, this subjectivity might influence the flavor ratings. Additionally, factors like the freshness of coffee beans or brewing time could introduce variability. Discussing these limitations demonstrates a critical understanding of your research process and helps identify areas for improvement. Recognizing limitations is essential for transparency and can lead to more refined future studies. Revise the experiment. Incorporate any feedback received during the peer review process to revise your experimental design if necessary. Adjustments may involve refining your methodology, changing variables, or improving data collection techniques. For instance, based on peer suggestions, you might want to explore different coffee to water ratios or modify the brewing process. Revising your experiment allows you to address any limitations and strengthen the reliability of your findings. This iterative process is essential in scientific inquiry, promoting continuous improvement and helping ensure that your research yields meaningful results. Reflect on the process. Take time to reflect on the overall research process, evaluating what worked well and what challenges you encountered. Consider how each step contributed to your understanding of the topic and the effectiveness of your methods. Reflecting helps you recognize areas of strength and those needing improvement, guiding future research endeavors. For example, you might note the importance of precise temperature control and how it impacted flavor extraction. This reflection fosters a mindset of continuous learning and adaptation, essential qualities for any researcher. Document everything. Maintain detailed records of all steps, observations, and outcomes throughout your research. Good documentation is vital for ensuring the reproducibility and credibility of your experiment. Keep a lab notebook or digital files that chronicle each trial, including dates, methods, and taste test results. This thorough record keeping allows others to follow your methodology and replicate your work if desired. Comprehensive documentation not only aids your own understanding, but also supports the scientific community by providing valuable data for future research and discussions. Stay curious. Cultivate a mindset of curiosity and exploration, recognizing that scientific inquiry is an ongoing process. New questions will continuously arise, leading to further investigations and discoveries. Stay engaged with your field by reading recent studies, attending conferences, and participating in discussions. For instance, explore how different brewing methods, such as siphon or pour over, affect flavor extraction from coffee beans. Remaining curious encourages lifelong learning and helps you adapt to new findings and advancements in science. This attitude fosters innovation and drives scientific progress. Conclusion: scientific inquiry enhances our understanding of the world. Exploring coffee extraction teaches essential research skills. Critical thinking and communication are key components. Curiosity fosters ongoing exploration and discovery. This process contributes to knowledge advancement in various fields. It equips us with valuable skills for everyday life. Thank you for your time and attention.